Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about complement systems. Okay, now in the previous series of video lecture, if you guys remember, we talked the physical barrier. Okay, now this is another series of video lecture to be going on, and in this video lecture we are going to talk about complement system. So in the series of video lecture we are going to talk about the complement system. Okay, this is the first video lecture, and in this first video lecture we are going to talk about the introduction, the basic overview, the big picture of the complement system. So let me write. Now, what is complement systems? Inside our body, if the pathogen or any microorganisms are get internalized, or let's say the bacteria or virus are get enter inside our body, then what actually happen? So after that, immuno responses are happen. So let me write. Okay, usually two kinds of immuno response are activated. One we call that humoral and other we call that cellular. So first of all, cellular immunities are activated. So let me write. And the other we have humoral. Try to remove the spelling mistake by yourself. Okay. Now, first of all, in cellular immunity, we already talked about the cellular and humoral immunity, but we just talked their basics. Okay, we not talk their detail. Okay, if you missed that video lecture, so it is present into my channel, you can watch it. Now, in cellular response, what actually happened? We have cells activated against the microorganisms. So what kind of cells we have? We already talked about, but let me write here just for your understanding. We have T cells. We have macrophages, dendritic cells, natural killer cells. Okay, these are the cellular response. Why? Because in this case these cells are activated, and once each cells are activated, then they can control the bacterial infection. Okay, now next we have another responses, we call it humoral response. And in humoral response, what actually we have a cells usually activated, we call that B cells. So let me write. This response is activated immediately. On the other hand, humoral response take days, it takes hours, it takes weeks for the activations. Okay, now during this response, first of all, B cells is activated. Now this B cells once activate, so then these B cells are converted into a cells which we call that plasma cells. Now plasma cells, let me write, they are converted into, and all of you better know that plasma cells are actually antibody machine, or these are antibody factory. Now this plasma cell produce what? They produce antibodies. Okay, these antibodies will attach on the bacterial surface. So after that they will attach. So let's say this is the bacterial surface. On the bacterial surface there are the antigen. So let me draw with the green marker. So let's say these are the antigen. Okay, so on the antigen such antibodies are attached. Let me draw with the black marker. So these are, let's say, antigen which are specific to, these are the antibody which are specific to antigen. Now what actually happen? Sometimes this antibody have the ability to kill the bacteria alone or by itself. Sometimes this antibody will call upon the phagocytes, and the phagocyte then engulf it, and finally these cells are killed. Okay, now after that, sometimes antibody, this is called antibody mediated pathway. Once the antibody are activated and they can kill the bacteria. Sometimes antibody mediated pathway are activated. After that we have another pathway which we call that complement pathway or complement systems. Okay, so let me write. So in complement system we have different kinds of proteins. We have almost we have 30 types of proteins. Okay, 30 types of proteins we have. Let me write. Enzymes, we have 30 types of enzymes or proteins. These proteins are sometimes glycoproteins, lipids, or glycolipids. Okay, but usually these are the proteins. Okay, now each of the proteins have two subunits. Let me draw. Let's suppose if this is the complement. Now these proteins are usually called as complement proteins. Okay, we call that C proteins or complement protein. Now this complement protein have two subunit. One is the large subunit and other is the small subunit. So this one is the small and this one is the large. Okay, this large subunit have the ability to attach on a bacterial surface, and this small subunit have the ability to release out, and they will act as a chemotaxis and call upon the phagocytes, and after that the phagocyte will perform the phagocytosis process. Okay, so first of all, what actually happened, this complement system is a very strong and powerful system as compared to this antibody mediated pathway. Okay, now what actually happens, sometimes each complement proteins once activated, so they are attached on the bacterial surface, and after that they will create a pores inside a bacterial cell. After that, not inside, means outside the bacterial cell surface, they create a pores. Now after the pores, the material which is present outside on the bacterial, the material which is present outside will be internalized, and the material which is present inside the bacteria will be released out. Okay, so what actually happened? The process is disturbed inside and outside of the bacteria. So sometimes the bacteria are swell up, burst. They are swell up, burst, and die. Sometimes these bacteria are shrink and die. Okay, so this is the phenomena of the complement systems. Okay, now if the pores are not formed, let's suppose the pores are not formed, so then the complement proteins which are attached on the bacterial cell, they, this bacteria are looking tasty for the rest of the immunocells, means phagocytes, then it is engulfed by the phagocytes. Okay, but sometimes these complement system are create the holes. Okay, now so this complement system was first of all discovered in the human blood serum. Okay, and this complement system consists of 30 protein, as I told you. So usually they have, if we talk the 30 proteins, so usually they have C1 protein, they have C2, C3, C4, C5, C6, C7, and we have C8, C9. Okay, now this C1 have three subunit. Name C1Q, we have C1R and C1S. These are the three subunit of this C1 protein. Similarly, this C2 protein have two subunit. So they have CA, C2A and C2B. Okay, this C3 has the same thing, C3A, C3B. Similarly, C4A, C4B. Okay, and so on, C5A, C5B, C6A, C6B, C7A, C7B, C8A, C8B, C9A, C9B. All these are make almost 30 proteins. Okay, so the nomenclature of each proteins are very difficult to understand, and it is very confusing, and it is very difficult to memorize each protein's names. Okay, now what are the action of each proteins are what? The main function of all these proteins, they are specially responsible to enhance phagocytosis. Okay, next we have this complement systems has three physiological activity. So let me write here. Now in first case, what actually happened? The first physiological activity, we have our body. So the first part is, or the first physiological activity is defending our body against bacterial infection. Now, what actually happened in this first physiological activity? In this case, in first physiological activity, the complement system act as a defending our body. They can defend our body against the bacterial infection. Why? Because they can facilitate and enhance the phagocytosis process, or they can enhance and facilitate the phagocyte cells. Now, how they can enhance and facilitate the phagocyte cell? They can, perform different kinds of processes. They can perform opsonization. Due to opsonization. Now, what is opsonization? First of all, let me a little bit explain. Opsonization, if you remember, we already talked opsonization, but let me once again tell you. Opsonization is that process in which the bacterial cell is coated by proteins, complement proteins, or it is coated by antibody or mannose binding proteins. Due to this way, this bacterial cell is looking tasty for the phagocytes. Okay, we call that opsonization, and then phagocytes can engulf it. All these phenomena are known as opsonization. So due to opsonization, chemotaxis. Now, what is chemotaxis? Chemotaxis is that phenomena in which the chemical signals are released, and such chemical signals are released from the T cells, and each chemical signals can activate the phagocytes, and then phagocytes can engulf the bacterial cell or any microorganism. We call that chemotaxis. So due to chemotaxis and due to the opsonization, so due to this processes, we can say that this complement system can defend our body against the bacterial infection. Okay. Next, we have this complement system can establish a bridge. Establishment bridge between innate and adaptive immunity. Now, how this bridge is formed? The bridge is usually formed that in innate immunity, the cells are activated. Means phagocytes, these kinds of cells are activated. And in case of specific or adaptive immunity, the antibodies are activated. The antibodies are attached. Okay, so that's how the bridge is activated between innate and adaptive immunity. So if I explain it with the help of diagram, so let's say if this is the bacterial cells. Sometimes these bacterial cells are, killed by these cells, which is actually the innate immuno portion, and sometimes it is killed by this antibody, which is actually the specific immuno system. Okay, so that's how we can say that the bridge is formed between the innate and adaptive immunity. Okay, now third physiological activity which are usually happen during complement system, we usually call that disposing of waste. Now the third is disposing of waste material. What does it mean? It means that once the bacteria are killed, so not only the bacteria are killed in this case, the cells which are activated in this case means dendritic macrophages, neutrophils, monocytes, all these cells are activated against the bacteria. So after that, each cells are die, means bacteria and WBC are die. So they can make a pus, means a pus is formed. Now after some time, each pus are then released out from the body. Now we call that disposing of waste. Okay, so these three physiological activity are usually happen in case of complement systems. Okay, so now let me once again explain the opsonization. Okay, now in opsonization, if you remember, we talked that it is, let me write here. In opsonization, we have what? We have antibody, we have complement proteins, we have mannose binding proteins. All these molecules are called opsonin. So let me write. So these molecules are called as opsonians. Now this opsonian, these molecules which we call it opsonians, they can create a bridge between the microorganisms and phagocytes. Okay, now how? Let's suppose if this is again, now let's explain this one diagram. Let's suppose if this is a bacterial cell, it is coated by the antibody, and sometimes it is coated by the mannose binding proteins. Let's say these red one represent the mannose binding protein, and sometimes it is coated by the complement proteins. So these blue one represent the complement proteins. Okay, due to this way they can create a bridge between the micro, now this is the microorganisms, and after that the phagocyte cells are activated. So let me draw with the red marker. So let's say this is the phagocytes cells. It may be neutrophil, monocytes, dendritic cell, or macrophage, any kind of phagocyte cell it is. So once it is activated, so then they will engulf it. Okay, so how the bridge is formed? Now this is the microorganisms and this is the phagocyte cell, and these are the opsonian. So opsonians are present in the mid area. So that's how the bridge is formed. Okay, so usually we have three complement systems, or we have three complement pathway. So let me write here. We have three pathway. We have the first one, we call that alternative pathway. Second, we have classical pathway. And third, we have MBL pathway, mannose binding lectin pathway. Okay, usually these two are very important for the exam and in our daily life. Okay, but don't worry, inshallah I will also explain this third one as well. Okay, so inshallah in the next video lecture we are going to explain this alternative pathway. Okay, I hope you understand about this video lecture. If you have any kind of questions in your mind, if there is something missing, so you can write it into a comment. Inshallah I will give you answer as soon as possible. If you like this video, so make sure to hit the like button, share this video, and also subscribe the channel, and stay tuned, because in the next video lecture we are going to talk about alternative pathway in a detail. Thank you so much.